I want to welcome you all here on behalf of the Discovery Centers for Eye Research and the Gavin Herbert and Eye Institute. This is our 6th annual translational research meeting, and which we've called a bench to bedside meeting. We're lucky to have it in this wonderful facility with its great food, and it just makes the meeting so much more fun, and we can interact at lunch and at coffee breaks. This is the 6th year that we've been doing it. It's skillfully put together by Dr. Christina Kenny, and the idea for this meeting came from Jack Scholerman, who is the chairman of the Discovery Centers for Eye Research, who's here today. Jack, we thank you for this idea. This has been wonderful. So we welcome you to a day that I think will be challenging and interesting, and perhaps you may even come up with somebody to collaborate with in your research. That has happened over the years. Some important collaborations have started. I'd like to ask Barry Cooperman, who is the professor and chairman of our department, to open the first session. Barry?
Well, I also wanted to echo Tony's words and thank everyone for attending, Jack for this great idea, Chris for all this incredible effort you've done in putting this together. Others have also been very involved, Dana Collinson and Jan Slavin in particular, and there's probably many others that I should name, but this in many ways is my favorite meeting of the year. It's really meant to encourage collaboration between scientists and clinicians. The bench to bedside is really meant to be this way. We have another very interesting meeting every year. This year, I think it's November 1 and 2, our clinical colloquium, which we tend to hold over at the Gavin Herbert Eye Institute, and you're all welcome to attend that as well. That's a similarly exciting meeting, but with a much more direct clinical focus. Again, the hopes of this meeting, as Tony has mentioned, is that not only we learn things that we didn't know were available both on campus and from the people that we've invited up from outside, but to encourage and foster collaboration. So please keep that hat on as you listen to everything today. And I think the speakers also have that in mind, how they can foster that idea. With that in mind, it gives me great pleasure to welcome our first speaker, Pierre Baldi, who is here from UCI, one of our own, very own professor at the Institute for Genomics and Bioinformatics in the School of Information and Computer Sciences, and he's going to be speaking about something that all of us are beginning to think about, but he's really doing a ton on AI, artificial intelligence, and deep learning, and how it applies essentially to all the biomedical sciences. But of course, we're all going to be thinking about it, how we can, we're so data-driven. We have so many images that we collect all the time in ophthalmology. Think about projects we could come up with once we hear words from Pierre. So thank you so much, Pierre. I really appreciate it.
Good morning. Thank you for the invitation to speak to you. This is a very exciting time for sciences in general. Just to give you a sense of how things have changed, this is you see here a picture of Galileo Galilei, who built one of the first telescopes. You see he's not very happy because he couldn't order it on Amazon. He had to build the telescope, and to thank him for his discovery, he went more or less to jail. The telescope had a magnification of a factor of 20, and of course he had to write everything on a piece of paper, and he had no storage devices and nothing to analyze the data but his brain, which was pretty good. But today, things look like that. You have the Hubble Telescope, the magnification factor is 5,000, and this instrument can beam petabytes of data down to Earth. And of course, you have to worry about storing this data, and most importantly, how to analyze the data. And that's where all these methods we have today in AI and machine learning and deep learning, etc., come into play. And you see this across all sciences and technologies. In biology, for instance, you look at microscopy, the first microscopes invented around the same time. And today, of course, we have all kinds of instruments to interrogate cells, DNA, proteins, etc., in very precise ways, and again, producing terabytes of data that we have to store and analyze. Same thing for imaging and all kinds of other modalities. The good news is that the techniques we have are actually very simple to understand. They are just a slight extension of something you know since high school, which is linear regression. So in linear regression, you have a bunch of points, that's your data, the blue dots, and you're trying to fit a model to those blue dots, and it happens to be a very simple model. It is a line. What we're doing today is the same thing, but we have slightly more complex models which are not linear, and we operate in very high dimensional spaces. But the idea is essentially the same. All the problems of machine learning can already be seen in this figure if you think about it carefully. So the models we use, they go back to the 1950s, where people started thinking about how the brain may be working, and in fact, all the basic concepts about neural function were already in place around 1950. They knew about synapses, evolution, action potentials, etc., etc. And so the idea, one idea that came up is to try to model a neuron as a simple processing device that consists a device that takes a weighted average of its inputs, which you see here. And if you stop here, it's just linear regression. You're back to a linear model. But then you pass this weighted average through a nonlinear function, for instance, a threshold function or a sigmoidal function. I mean, there are different such functions, and you produce an output. And this very simplified model of a neuron is basically what we're using today in deep learning. We're just building large circuits of devices of this type, and what we call learning is simply how you store information in the weights of these processing neurons so that they implement the right input-output functions. In particular for images, around in the late 70s, Fukushima came up with what is called today a convolutional neural network, a neural network for computer vision, for image processing, which was very much inspired by the work of Hubble and Wiesel in the 60s, where they studied the primary visual areas in cats and discovered that there were feature detectors, that is, neurons that could respond, for instance, to oriented bars at specific locations in the visual field. And so the idea is to build an architecture which is very much inspired by the work of Hubble and Wiesel, where you have an array of receptors, where all the neurons within one array are applying the same operation, for instance, detecting certain bars at certain orientations. And you stack this layer up, and you use this architecture to process images. The only problem is that Fukushima at the time said that in order to learn the value of the weights between the layers, he proposed to use Hebbian learning, which is another idea that goes back to the 1950s, and nobody was ever able to get this to work. In fact, 5 years ago, we proved that it's impossible to have an architecture of this kind capable of processing images and detecting interesting things if all you need is Hebbian learning. You need something else, which is a channel, which we call the deep learning channel, that is capable of providing information about the performance of this architecture back into the deep layers and the deep synapses. And basically, this is what gradient descent does. So gradient descent was introduced also in the late 70s, early 80s by various people. And the basic idea is that you have some kind of error function at the output layer that tells you how good your performance is. So you put an image, let's suppose you want to recognize cats in images. You put an image here, you propagate forward through the network, and here you get a certain error depending on the output of the network. And then you do gradient descent to try to reduce this error. There is a way of computing the gradient of the error with respect to all the weights between all the layers. And you just change the weights by a small amount in order to reduce the error. And you keep doing that. It's called stochastic gradient descent also, because you do this example by example, so there is some stochasticity in the descent process. But that's all there is. All the things you see today, self-driving car, et cetera, are based on this very simple idea of using gradient descent to reduce the error. And so in the early 90s, late 80s, early 90s, we started getting these techniques to work on images. And so one of the first applications to biomedical data, or in this case, I would say biometric data, was a convolutional architecture of the style of Fukushima applied to fingerprint images to detect whether two fingerprint images correspond to the same finger from the same person or not, for instance. And today, basically, the basic ideas are the same. This is what is being used in deep learning across all areas of science and technology, but the main changes are the computational power. We have GPUs, which we didn't have in the 80s. The data, for many applications, you have very large amounts of data that are available, in part due to the internet, databases, etc. Facebook, Google can easily access 1 billion examples of images of human faces, for instance. So the data, the computing power, and of course, now we also have very, very good software for implementing these techniques, because companies like Google and Facebook are making available their software publicly. So you have libraries like TensorFlow, PyTorch, Keras, which allow you to deploy these techniques very rapidly now. I routinely have undergraduate students in less than 6 weeks being able to implement and deploy these things very, very effectively. And in many cases, for specialized problems, you can achieve performance that is at the same level, on par with humans, and sometimes better. So you hear about systems that can play Go better than humans, play chess better than humans, recognize images better than humans. It's all driven by this kind of technologies. So application to the biomedical sciences, you can apply these techniques. It's like linear regression. You can apply linear regression to anything. It doesn't matter if it's data coming from psychology, from medicine, from physics. It's the same technique that you apply across those different disciplines, different kinds of data. Same thing you see here. You can apply deep learning to problems in chemistry, problems of small molecules, drug discovery, etc. You can apply to protein, genomic data, gene expression data, and of course imaging, which is probably the most interesting for ophthalmology, but beyond that also to electronic medical records, to text data, etc., etc. So this is, I'll just show you a few quick examples before I move to imaging. This is an example where we predict the properties of small molecule, whether a molecule is soluble or not, which is important in drug discovery. So you have neural networks. I won't go into the details, but they are basically neural networks that are crawling these molecules and producing an output, in this case predicting solubility. And again, you can compute an error here and then use that to adjust all the weights in these architectures. Many of you are very fond of organic chemistry, so predicting chemical reaction, bromination of alkenes. We do this with deep learning now. We're trying to get rid of those thick books that you had to memorize. Basically, a reaction, if you go back to basics, it can be described in terms of arrow pushing mechanisms, where you are trying to identify sources and sinks of electrons in the reactants. So we do this with neural networks, find what is the best source, find what is the best sink. Of course, you may have several sources and sink, so you go look at all possible source-sink pairings, and then you have to rank them. And ranking, again, is a standard thing, is machine learning. That's what Google does when you enter a query. You get a ranked list of hits. So we use, again, what is called a Siamese architecture to compare possible source-sink pairs and identify what is the optimal source-sink pair to implement the reactions. This is another example of an architecture for analysis of genomic data. I don't have time to go into details. These architectures are very similar to those that are used for language processing. You should see Google Translate, translation from English to Japanese, etc. It's all done with this kind of techniques for processing text, which are coming straight out of what I described. Protein structure prediction, for instance, this is called a bidirectional recursive neural network, where you have the protein sequence in the input, and here in the output you would like to have, for instance, the prediction of the secondary structure, where are the alpha helices, the strands, and the coils. And just to give you a sense of how the performance has improved over the years, the first neural network application to secondary structure prediction, it goes back to the 1988 by Kian and Sinowski. The accuracy was about 64%, and now we're up to about 95% for predicting secondary structure, and this is probably as high as you can go for a variety of reasons, because there is noise in the databases, there are proteins that do not fall spontaneously, disordered regions, et cetera, et cetera. Again, very quickly, this is an application, again, of deep learning to omic data, where we're trying to identify transcript, for instance, or metabolites that behave in a circadian way. So you do gene expression experiment, RNA-seq experiment, for instance, every 4 hours over a period of 24 hours to measure the concentration of transcripts. And then you are faced with the problem of deciding which transcript are oscillating in a circadian manner. So this is very important for the study of circadian rhythm. We're collaborating with the laboratory of Paolo Sassone Corsi on these problems. So let me get to images, which is probably the most relevant for applications in ophthalmology. And again, I'm going to give you examples of image processing problem in a variety of applications within medicine, and also at different scales and different imaging modalities. So this is Chris Hughes in the School of Biological Sciences. He has developed a microfluidic system where he can grow blood vessels in these little chambers that you can image. And of course, you can also, more importantly, grow cancer cells. The blue that you see here, these are cancer cells that are growing, and you can, of course, give drugs. You don't want to stare at these images. You can get hundreds of images per hour. What you're interested is to detect whether the vasculature is changing, whether the cells are growing or shrinking, etc. And again, this can be done very effectively with these techniques. This is another example we're just starting to work on about drug delivery. So the shiny things you see here are nanoparticles. These nanoparticles are penetrating cells. This is a project. The goal of the project is to develop nanoparticles that can deliver drugs inside cancer cells. You can see cells here. And again, you can get a lot of those of images of such type, and you want, for instance, to be able to detect whether the nanoparticles are inside the cells or outside, whether they are delivering drugs or not, and all that can be automated. Another example, collaboration with Sabi Molloy here in radiology, where we're looking at mammograms. And of course, one task is to try to predict whether there is cancer or not in the mammogram. But here we're doing something else. We're looking at the vasculature and the degree of calcification. High level of calcification is a potential sign of heart disease. So as a byproduct of being screened for cancer, you could also be screened for heart disease. And you can see here the regression line between the predicted level of calcification and the ground truth provided by radiologists that have looked at these images and spent time trying to estimate the level of calcification. You see that there is a very good agreement. Another example, metastasis, detection of metastasis to the spine. Again, same thing. You can use this convolutional neural network to detect whether there is a metastasis or not in the given image, and if there is metastasis, where is it located exactly? I'm sure that I think Peter is here, will tell you about numerous applications to the brain, so I'm not going to say much about this. Of course, there is work going on in other places. This is an example of a project where essentially people can take images with their cell phone. They can take an image of a skin mole, and the image can be sent to the cloud, and there is deep learning that processes those images and can detect whether there is melanoma, or at least potential for melanoma, and send back a recommendation whether the person should go see a doctor or not. In dermatology, these are experiments where we're trying to automatically count the number of a hair. It sounds like a trivial or silly problem, but it's actually important in dermatology, because if you have a drug and you claim that the drug is capable of promoting hair growth, how do you really prove it? The only way is to count how many hairs, follicles there are in a certain region, administer the drug, and then keep counting in the coming weeks, right? And that's very difficult and very tedious. So we're trying to automate that process. And what I've shown you already gives you a sense of the breadth of applications you can do and the different types of imaging modalities. Some of you may wonder whether this can be applied also to videos, for instance. And I'm going to show you that yes, it can be applied to video. So this is an example of a project done with Dr. Kearns, which I think is speaking later, about detecting polyps in colonoscopies. So we had a few thousand images of images of the colon with and without polyps. And so we can train an architecture to predict whether in a given image there is a polyp or not. And in fact, we can also predict, if there is a polyp, we have methods that allow us to draw bounding boxes around the polyp. Now, if you have video, you may think that this is not enough. It's not sufficient to handle video. But the point is that in order to train these neural networks, the training process may be long. It may take 1 or 2 weeks on a cluster of GPUs. But once the system is trained, it is very fast. It takes about 20 milliseconds to make a prediction. So if you have a video, we just take frames, maybe not every frame, but every 5 frames or every 10 frames, we make a prediction. That is plenty. And I'll show you an example. Here is the video. You will see an example, some clips from various colonoscopies, and you will see polyps in some of the frames, and you will see our deep learning system that is putting bounding boxes around the polyps in real time. All this is done in real time. Okay. And again, you can apply this to other kinds of data. This is a project done with Maxime Canesson, who is in anesthesiology, was here now, he's at UCLA, where we're trying to predict post-operative outcomes after surgery, in particular, for instance, things like kidney failure, given a number of vital signs and measurements that are done before and during the operation. Again, using the same techniques. And so moving to ophthalmology, where I must say we haven't done much yet, but we've started to talk to Ken and Andrew and Charlotte about all kinds of interesting problems. For instance, here is an idea from Ken, which is to process images in order to identify the TM, which is very important in some surgery. You can see here, it's this, I'm told it's number 3 in this picture, or here. So all I have is to detect where's number 3 in the pictures, right? So that's an example of things we're starting to think about, among other things. So some conclusion, these deep learning methods are very effective and very flexible. They can apply to all kinds of data and all kinds of technologies and sciences. Convolutional neural networks in particular are very useful for processing, analyzing, mining images, and this can be done across different types of images and sensors. It doesn't matter whether it's microscopy, X-ray, ultrasound, etc. We can deal with all these kinds of images in the same way. Of course, the size of the image, if you have very, very large images, is going to have an impact on how fast we can process them, etc. But basically, all of those can be analyzed. The key really is the data availability. Every time we talk to doctors and we ask for images, for examples, there is always some hesitation, then it takes some time to get the data and how much data we can get. All those things are really, as far as we are concerned, the main problem, the main obstacle that has to be overcome. One point that I want to stress is that we used to need a lot of data, and of course the more data we get, the better, but there are all kinds of techniques today that allow you to work with relatively little data. So there are things like data augmentation, where from a single examples we can produce many examples by doing things like translations, rotations, adding noise, etc., to expand the training set. There are techniques for preventing overfitting. If you don't have a lot of data, one of the big dangers by using these models with a lot of parameters is overfitting. So there are all kinds of techniques for regularizing the problem, for avoiding overfitting. And perhaps the most important thing is that we can use networks that have already been trained. This is a technique that is becoming very popular. You take a neural network for vision that has been trained, let's say, for a self-driving car. So it's a network that can recognize bicycles, pedestrians, and et cetera, right? And it's a very big architecture. Now, most of the layers in that architecture are general purpose vision. They are detecting edges like Hubble and Wiesel, etc., right? So all those you can still reuse, you just have to retrain the top for your specific task. So if you are interested in detecting the TM, for instance, I could take one of those architectures that are used for self-driving car and just chop off the top, put some new neurons there, and train the top for the TM problem. You're still going to need the lower layers that are doing edge detections and things of that sort. There are methods for compressing these models, in particular applications where you are hardware constrained. If you want to put these neural networks, these big neural networks into cell phones, we have techniques for compressing those. And in a sense, you may get the impression that all the problems are solved in computer visions, and it is a little bit true. I think all the biomedical imaging can be addressed with current techniques. There are still, in some cases, some subtle problems that we're still working on. But the basic problems of classifying images or creating bounding boxes around regions of interest, all those things are very doable in various settings as long as we have enough training data. There is the black box problem. Some people are worried about. You train these networks, they may perform very well, but you don't know how they do it. So that's the black box problem. It is a problem. There are techniques for understanding what the network is doing. They are very time consuming. But I think the real solution is in a number of techniques like encapsulation. That is, you take a problem, you break it up in different sub-problems, and you have neural networks or other techniques dealing with one of the subproblems separately, because that allows you to rapidly identify where are the points of failure and how to fix them. For instance, in the colonoscopy problem, if you train a network just to tell you yes or no there is a polyp in the image, let's say the system makes a mistake, says yes, there is a polyp and there is no polyp in the image. You have no idea of why the network said yes. But if you do the classification problem plus the bounding box problem, now you have a very good tool, because the network says yes, it makes a mistake, but it also tells you where it thinks the polyp is. And so you can go into that area, and maybe you will notice that there is a surgical instrument. And then you understand that every time there is a surgical instrument in the training data, the system has learned to make the inference that there is a polyp in that image, and it's not correct, of course. But you can fix it by fixing your training data, for instance. So encapsulation is very important. And in closing, I think the main challenges are collecting data, sharing data between the biomedical community and the computer science community, and of course, then you have the problem of deploying this technology in healthcare settings, which also raises a number of interesting issues that perhaps can be discussed during the break. So I think there are many opportunities for applying these techniques to ophthalmology. There are plenty of students in computer science who are very interested in working on biomedical problem. And so the key things is to build the bridge and find ways of exchanging data in ways that are efficient. And of course to collaborate so that it is absolutely necessary to have teams of scientists from both sides that can talk to each other in order to develop efficient applications. Thank you. Yeah, of course, those are good questions, and the answer is yes, people are working on those, not necessarily just my group. But so the first question is, secondary structure is really the first step, right? Then, to go to these 3D structure, you predict contact maps. So we go from secondary structure and other things like solvent accessibility, we predict contact maps, and then from contact maps, we go to tertiary structure. That's the pipeline. And you can bring evolutionary information. There is a lot of work going on those kinds of things. And at the last, there is a CASP conference where people benchmark methods every 2 years. These methods, machine learning methods, deep learning, are doing the best. And of course, you can do the same thing or start doing the same thing on membrane proteins. The problem is you have fewer examples, because it's much more difficult to deal with membrane proteins, to crystallize them and get their 3D structure. So in the PDB database, where you have your training examples, 95% or more of the examples are globular proteins, are not membrane proteins. Well, that's certainly not going to be my decision. That's going to be the decision of the MDs, right? So I think, of course, you compare it to human performance, and we see over and over that these things, when they are well trained, they become better than human. They have superhuman performance, and it's not surprising. So I think, if you look at the trend, that's the long-term trend. I think all this thing is going to be handled by machines. But when you decide that it's good enough or better than humans, you have to compare to humans, basically. Well, definitely AI is meeting medicine. I think one of the things that you're pointing to is the issue of rule-based systems. Symbolic systems, which were very popular in AI in the 70s, etc., in some cases are still very good, like 3-SAT, satisfiability solvers are becoming very good. And so what is the role or the interface between this rule-based system and this more neural connectionist architecture? So I think that's one of the fundamental problems, open problems in AI, is what is the connection between those two, how you combine them together, how do you leverage one to help the other, et cetera, et cetera. So those are, beautiful and very interesting questions. And I don't think we have answers to those questions yet. However, one should be cognizant of the fact that there must be solutions that are from one end to the other neural, because that's what the brain does. Right? So there must be a way of implementing rule-based system through a neural architecture. And I think that's the direction that interests me personally the most. Again, with this method, you need training data. So rare diseases depends on which disease you're thinking of and which data, right? Is it the genomic data? Are you looking at images of a particular organ, etc.? So it's going to depend on which modality you're using for the diagnostic of those diseases, and then how much data do you have, and how distinct is it from the background, right? So I think there is no generic answer. It's going to be disease specific.
All right, thank you, Pierre.
Thank you.